So today, my name is Oliver Hatley, and I'm a principal scientist at Certara. And this webinar, the Certara webinar, is here and organized originally part of the SimCip webinar series, where we look at real-world case studies showcasing the application of the SimCip simulator. So you can see past talks on the Certara website, on the webinar website, and on YouTube. As you'll see in the presentation today, there is a significant role of PBPK in model-informed drug development and its role in answering key regulatory questions and allowing for clinical trial study waivers for drug product labeling claims. So today, we're very lucky to have a talk by Dr. Ioannis Lossius Konstantinos, a principal scientist in the modeling and simulation group at Novartis. And the title of his talk is Physiologically Based Pharmacokinetic Modeling and Simulations to Support asciminib NDA submission and inform Scemblix's drug product label. So Ioannis, without further ado, thank you for agreeing to speak to us today, and I'll hand over to you to give our presentation today.
Thanks, Oli, for the kind introduction, and thanks the SimCip and Certara teams for inviting us today. And hi, everyone. So, as Oli mentioned, today we are going to discuss about the impact of physiologically based pharmacokinetic modeling and simulation to support the asciminib NDA submission and to inform Scemblix's drug product label. So if we go to the next slide, here this slide shows that Scemblix, or asciminib under the trade name of Scemblix, was recently approved by the FDA in October of 2021, and earlier last year in March and August by PMDA and EMA respectively. Now, if we look a little bit at the path towards the approval of the drug, we had, of course, one phase 3 trial, which was supported by the NDA submission to both FDA, EMA, and PMDA, and particularly for FDA, which granted us a priority review and accelerate the approval for orphan drug disease designation under real-time oncology review. We also had with FDA two FDA breakthrough designations, and particularly for the USA, even though only one dose level was tested in the phase 3 trial, 3 dose levels were approved by the FDA. So the 40 milligram BID in third line CML, where we had also the phase 3 trial, and additionally to it 80 milligram QD and 200 milligram BID, particularly for patients with the T359 mutation. And we will look a little bit more on the approval of these 2 additional doses in the upcoming slides. For the 8 and 200 milligram doses, and for the approval, instrumental role played PBPK modeling simulation, as well as support by population PK and PK/PD analysis. So we can move to the next slide now, where we are going to talk a little bit more about the mechanism of action of asciminib to treat CML. So as you may know, CML is a malignant hematological disease characterized by the leukocytosis, and from the Philadelphia chromosome positive CML, where we have is caused by the reciprocal translocation of the chromosome 9 and 22, producing the Philadelphia chromosome, which is a smaller chromosome 22, which resulting into the fusion of the BCR-ABL protein. So if we look particularly at the mechanism action of asciminib, unlike other tyrosine kinase inhibitors, asciminib is specifically targeting the ABL myristoyl pocket via a mechanism called STAMP. Now, looking at the drug and human PK properties of the drug, asciminib, it's a BCS class 2 weak base, which is formulated as the hydrochloride salt. It exhibits pH-dependent solubility, and in terms of human PK, the Tmax of the drug is around 2 to 3 hours at all dose levels. There is no time-dependent PK, meaning that there is no time-dependent inhibition or induction of the drug itself. And also the drug is highly bound to proteins. The volume of distribution is relatively small. The main elimination pathways involved, and we'll look into a bit more into detail into that, is CYP3A4, BCRP, and different UGTs, including UGT2B7, UGT2B17, UGT1A3, and 1A4. It's inhibitor of CYP2C9, CYP2C8, CYP3A4, and PGP. And also, it has been observed a significant reduction of the drug exposure with the presence of food. So if we go to the clinical pharmacology study package for the drug, these slides showcase the studies that we had available at the time of the NDA submission. So of course, this included a human ADME study with radio-labeled material, hepatic and renal impairment studies at 40 milligram, and 3 drug-drug interaction studies. The first where asciminib was given at 40 milligram twice daily dosing as a perpetrator with CYP2C9, CYP2C8, and CYP3A4 substrates, and 2 other DDI studies where asciminib was as an object with strong CYP3A4 inhibitors and inducers, as well as a PGP inhibitor, and lastly, a pH-mediated interaction where asciminib was co-administered with a proton pump inhibitor. In addition to this, there were relatively bioavailability studies and food effect studies, and also other clinical PK was available from phase 1 studies in patients in the dose range of 20 to 400 milligram total daily dose, as well from the phase 3 study at 40 milligram BID dose. Most of these studies have been already published in the literature, so if you're interested in the clinical pharmacology package, you can look at the publications from the colleagues that are listed here below. So one of the main questions that we had to address before filing, actually, and at the late stage of the development, was whether the 400 milligram BID dose was actually the right dose, comparing to the competition, but also for patients' compliance. As I mentioned before, food significantly reduces the exposure of the drug, which means that administration in fasted state, especially with a twice daily regimen, might complicate and the administration and reduce compliance. And on the other hand, already we knew from the phase 3 trial that there is a superior efficacy versus the standard of treatment, which was bosutinib when asciminib is administered at 40 milligram BID doses. So what we wanted to explore was whether a more patient-centric dose regimen of 80 milligram QD would be appropriate and could be justified. So if we click next, the challenge we had with this 80 milligram QD dosing regimen that we would like to propose was that we didn't have any phase 3 data. We had only some limited data from 18 subjects from phase 1, and also there were not clinical pharmacology studies available. So let's see how we address that question. So practically, there were 2 avenues here that we could have explored. In a classical way, that would be to run another phase 3 trial with a new dose level, which of course would be time and resource consuming, and also we would delay significantly the approval and I mean, apart from the approval, also the accessibility to CML patients. So instead of that, Novartis will propose to use a model-informed drug development approach to bridge between the 2 regimens and to allow to bring the regimen hopefully faster to the patients. And for that, we had mainly to address two main points. One was to bridge the efficacy and the safety between the two dosing regimens, and there is an interesting poster presentation by our pharmacometric colleagues on this topic. And the second was to use PBPK modeling to bridge all the DDI clinical assessment as well as the organ impairments assessment at other dose levels that the studies haven't been performed before. And this is what we did actually. If we move to the next slide. So this slide shows what we first started exploring, which was looking at the exposure between 40 milligram BID and 80 milligram QD, which, as you can see on the right part of the slide, the exposures overlapping. So if we look at the average steady state AUC over 24 hours, it was very comparable between the two regimens. And as expectedly, the steady state Cmax and Cmin between 80 milligram QD and 400 milligram BID were different, with Cmax being 1.6 fold higher and Cmin being 0.7 times fold lower comparing to the 40 milligram BID. And of course, further to this, using these PK metrics as well as the exposure of the drug, model-based exposure efficacy and exposure safety analysis was used to support our arguments of the similarity between the two regimens, and actually that the two regimens can be interchangeable. So let's now take a closer look at the label, and especially for the drug-drug interaction. So if you click next, you will see here on the section 7 of the USPI of the drug product label, you can see a clear mention of drug-drug interactions at 200 milligram and 80 milligram. So the labeling language here was significantly affected by PBPK simulations, since we didn't have any clinical pharmacology studies at the higher dose of 80 milligram and 200 milligram QD. And on the right part of the slide, you can see the specific labeling language for strong CYP3A4 inhibitors, for example, or other CYP substrates, as well as for PGP substrates. So now we'll take a closer look at the PBPK model development itself. And essentially the PBPK modeling strategy we implemented here is sort of a standard PBPK modeling strategy, starting with a model building of an initial PBPK model from in vitro and either preclinical or even clinical PK data when talking about human, and also starting with the development of a healthy adult PBPK model, which was based on the 40 milligram single dose PK data that we had from phase 1 studies. In the next step, the model was refined by simulating the PK in patients after single and multiple doses in a large dose range covering 20 to 400 milligram total daily dose, with either 20 up to 20 milligram, up to 200 milligram BID. And then the model, when it was able to capture appropriately the exposure with the dose, also there were further model adjustments that were performed in order to calibrate our model to predict the drug-drug interaction with CYP-prop substrates at 40 milligram BID. So on the next step was to verify the model by confirming and verifying the relative contributions of the elimination pathways, for example, for CYP3A4 with a strong drug with a drug-drug interaction study with a strong CYP3A4 inhibitor, and also verify the ability of the model to predict the PK with a training dataset of clinical PK data, as well as to predict the drug-drug interaction magnitude with CYP probe substrates. And of course, on the last step, and most importantly, once the PBPK model was established, was to apply the model in order to simulate untested drug-drug interaction scenarios for either CYP, UGTs, or transporters that were involved, as well as to predict the impact of organ impairment, either hepatic or renal impairment, at higher doses other than 40 milligram, and use that as a justification, as argument during our NDA submission package. So this slide shows you that, of course, when we're talking about PBPK modeling that supported the NDA submission, this does not mean that it's a one-time activity, but rather a marathon here, as you see, of more than 10 years, starting early from on the upper part of the slide, even at the preclinical development, looking at formulation optimizations, even in animal species, looking at trial designs, co-medications. And then, of course, over a longer period, the model is refined and re-verified with clinical PK data, phase 1, phase 2 data, until reaching the phase 3 and before submission levels, where we have finally the development of a final PBPK model on the left part of the slide, which was eventually submitted to the FDA. And since 2021, the impact that the PBPK model had on the drug product label, as well as, for example, additional post-marketing requirements and post-marketing commitments, where FDA asked us to perform using PBPK modeling, and other type of activities. For example, for us, it was the first time that we submitted e-data of PBPK models in Japan. So this clearly shows that it's a very long process that started many years ago. Now, one important component of the PBPK model verification and development is, of course, to define the relative contributions of the enzymes involved to the total drug clearance. And this started already, as you can see on the leftist box of the slide, already in 2013. Initially with in vitro reaction phenotyping and kinetics performed in human liver microsome in the presence or absence of NDPGA or UDPGA, as well as by defining human clearance in hepatocytes, and this gave us a first kind of estimation of the relative contribution between oxidative metabolism and glycuronidation, which you can see here with about 35% of CYPs and 60 to 65% of UGTs. Then on the next step, with additional in vitro experiments, we were able to define relative contribution of the individual CYPs enzymes from recombinant CYPs, as well as the contribution of the individual UGT enzymes with UGT in chemical inhibition and UGT with specific metabolites in hepatocytes. And from there, we're able to see that from the CYPs, CYP3A4 is mainly involved in the metabolism of asciminib. And then there is a mixture of contribution between different UGTs, primarily from UGT to B7 and followed by UGT to B17, and then UGT1A3 and 1A4. And now only in 2017, where we had the results of a human ADME study, this gave us a little bit of complexity on explaining and determining the elimination pathways. This was because 57% of the parent drug was found in the feces, and this could have been coming either from unabsorbed material or from active secretion or back conversion of the glycuronide to the parent. Now, with additional analysis that was performed using late fecal samples, about 24% of this could have been attributed to either secretion or back conversion of the glycuronide to the parent. And here you see below with red in the third box the two scenarios that we end up having from the human ADME study. So in the first case, if the 57% of the drug was coming from the unabsorbed material, would have 64% of CYPs versus around 30% of UGTs, while in the second case, where 24% comes from secreted or back converted secreted drug or back conversion of the glycuronide to the parent, we have about 37% of CYPs and around 58% of UGTs. And since this first scenario was more in line also more in line with also our in vitro assessment at the time, this what we were thinking it was the more likely scenario. Now 2 years later in 2019 we had a clinical DDI study with a strong CYP3A4 inhibitors, and from there we kind of confirmed that the percentage of contribution of the CYP3A4 was in the 30% range based on the results of the clinical DDI. And with regards to the human ADME, there is an interesting publication which is linked on the slide, so you can have a look for more details. So now let's move into the initial boulder building, where here you see an example on the left of some of the figures of the model performance at 40 milligrams single dose in healthy subjects. And the PBPK model initially was verified at 40 milligrams single dose to predict the PK in healthy volunteers from different clinical trials in 6 different cohorts and 4 different trials. And all the PK parameters were predicted reasonably well within 28% of the observed values, and as well as the inter-subject variability of the PK was also reasonably well captured. So once we had that, we moved on to further model refinement. And this was also complicated because the exposure of asciminib with dose follows a slightly over-proportional increase, so had to implement a nonlinear PBPK model. And to do that, our hypothesis was that this was coming from saturation of the BCRP in the liver, and this was because the in vitro constant of BCRP from the in vitro kinetics was about 10-fold lower compared to the Km values of all the other enzymes, both CYPs and UGTs. And additionally to that, by doing sensitivity analysis on the Km of the different CYPs and UGTs, we were able to show, or it was kind of observed that there was little or no saturation of these enzymes, even if their in vitro Km was reduced an additional 100-fold from the in vitro value. So this was our main hypothesis where the nonlinearity of the PK was attributed. And as you can see on the graph on the right of the slide, with the red, you can see how the PBPK model was simulating the AUC dose normalized versus the dose when the BCRP was not included. So practically we have a linear PBPK model, and how it does compare with the observed dose normalized AUC with the dose, which here with blue, and with the green line you see how the model performed when the BCRP was included and the nonlinearity was built, and you can see that it can approximate the observed PK data, or at least the observed dose normalized AUC much more reasonably than before. Can we go back to this previous slide? Sorry. Another thing I wanted to mention was the challenges that we faced in developing a nonlinear PBPK model, which included the use of the permeability limited liver model, and how we can perform an in vitro extrapolation of the clearance PD value, as well as further optimization that we had to do in a top-down fashion for the Km of BCRP by an additional 10-fold, in order to be able to better predict the PK of asciminib. But the final PBPK model, which was non-linear, was able to capture the PK parameters at day 1, day 15, and day 21 after single and multiple dose PK in healthy volunteers and cancer patients within 45% of the observed values, which was considered a reasonable good model performance. All right, so let's move on further with the model refinement. So the slide on the left, the figure on the left shows practically the model verification with the external dataset that was used once the nonlinearity was built, and how the model performs at different dose levels in cancer patients at 40 milligram, 80 milligram, and 200 milligram at day 1, day 15, and day 28. And the graph on the right shows additional sensitivity analysis that we had to perform in order to match, for example, the DDI, the drug-drug interaction with sensitive CYP substrates. For example, here you see an adjustment that we had to do to our in vitro KI for CYP2C9 that had to be reduced in order to predict the in vivo effect with the warfarin DDI. All right, let's move to the next. Now coming to the final step of the model verification, which included the confirmation of the fraction metabolized by CYP3A4. As I mentioned before, we had a clinical drug-drug interaction with CYP3A4 inhibitors and inducers. And we had with two strong CYP3A4 inhibitors, clarithromycin, as you see, and itraconazole administered as a capsule. And as a strong inducer, we had rifampicin. So from the interaction with clarithromycin, the results which were observed were more or less in line with the human ADME results, as well as the in vitro phenotyping data, showing a 36% increase in the exposure of asciminib, leading to a fractional contribution of CYP3A4 in the range of 30%. Now, on the other hand, when we looked at the results with the itraconazole capsule, practically we saw no effect, with only 4% increase in both Cmax and AUC. However, the PBPK model was calibrated based on the clarithromycin DDI results. Now, looking at the results with a strong CYP3A4 inducer with rifampicin, we saw only a reduction of about 15% in the exposure of asciminib in the presence of rifampicin, and a 9% increase in Cmax, which these results at the moment it was, and still until today, it's hard for us to interpret. But as I mentioned before, since the PBPK model was calibrated based on the clarithromycin DDI data, eventually the PBPK model overpredicted, as you see here with the magenta arrow indicated, overpredicted the induction effect by rifampicin in about 30%. So overall, our basic assumption, and we can go to the next slide, was that the FM CYP3A4 is about 30% and calibrated using the clarithromycin DDI data. The slide shows here the performance of the model in predicting the different drug-drug interaction studies for both AUC ratio on the left and Cmax ratio on the right, based on the GEST criteria and 20% of intra-subject variability. And as you can see, the model was able to capture all the drug-drug interaction studies within the guess criteria with a slight overprediction of the strong CYP3A inducer effect with rifampicin, which I also mentioned before. So this brings us to what we believe until today is the final contributions of the elimination pathways of asciminib, and as also has been implemented into the PBPK model. So there is a biliary secretion via BCRP, which contributes significantly to the excretion of asciminib, and also asciminib is mainly metabolized by CYP3A4 and UGT2B7. The absorption is expected to be complete, and the whole dose range of 40 to 200 milligram in the fasted state, and then there is about 25% of first pass metabolism in both guts and liver, which leads us to a predicted bioavailability of about 17.3%. And here on the right, you see the standard pie chart indicating the different contribution of all the individual enzymes and transporters. Now, for the contribution of BCRP, this was indirectly determined by switching on and off the BCRP in the liver at the dose level where we thought that BCRP is not saturated, and then calculating the percentage of the clearance that BCRP contributes, and this was then further subtracted by the contribution of the UGTs to account for the active secretion of the parent drug. All right, so coming now to the model application, this slide practically shows the untested, this slide and the next one slide show the untested drug-drug interaction scenarios that the established PBPK model was applied. So for example, and let me guide you through the slide, the verified model was used to predict the drug-drug interactions of CYP substrate at the two other marketed doses, meaning at 80 milligram QD and 200 milligram BID. So this is what we look at the forest plot on the left part of the slide. So this forest plot practically shows with the solid green line shows the presence of an actual clinical DDI. So here is the case for these three drugs, for these three substrates, midazolam, varfarin, and repaglinide as CYP3A4, CYP2C9, and CYP2C8 substrates, and then the dashed green line show the verified predicted by the PBPK model at the same dose, and lastly the red line here, dashed line, show the predicted by the PBPK model without the presence of any clinical PK data or any clinical DDI data. Now, you can see also the magnitude of the drug-drug interaction inside those boxes for both the Cmax and AUC ratio. And on the right part of the slide, the model then was that was calibrated, as we mentioned before, with the clarithromycin DDI data was used also to predict the DDI with other strong CYP3A perpetrators at a 2 additional dose. So here, for example, you see clarithromycin intraconazoles as CYP3A4 inhibitors and rifampicin as CYP3A4 inducer. Now, in the next slide, we have additional applications of the model, and here there are like cases where we didn't have any clinical DDI available. That's why you see these boxes being all signified with the red dashed line. So, for example, we had the weak DDI effects predicted for asciminib with moderate CYP3A4 inhibitors and inducers, which is shown here. And also there was predictions, PBPK predictions with other CYPs, UGTs, and as well as PGP substrates. For example, there was no 2-week DDI predicted for UGT1E1 substrates, here with raltegravir, and CYP2C19 with omeprazole and CYP1A2 with caffeine from 40 to 200 milligram. And also there was weak to moderate DDI effects predicted once for selective PGP substrates once the in vitro in vivo extrapolation for the PGP inhibition properties was established. So just as a side note, there will be another slide just after that explaining that, but originally our PBPK model did not predict any effect with PGP substrates. So we can move now on some of the highlights from the FDA summary of clinical pharmacology and PBPK regulatory review. So this coming to a question we had from FDA with regards to in vitro in vivo extrapolation of PGP inhibition by asciminib. So the information request by FDA was asking us to establish an in vitro in vivo extrapolation for the PGP inhibition properties, since it has been observed in the literature that when the in vitro KI is used directly into the model, there is a systematic underprediction of the clinical DDI effect. And indeed, this is what you are looking at the left part of the slide, where this graph shows how the model predicted when the observed clinical DDI effect of 15 different perpetrators with digoxin as a sensitive PGP substrate. And what you can see here is when the in vitro KI is used directly for these different perpetrators, where the inhibition properties were measured under the same lab and under the same conditions and same vesicle assay, you can see that indeed the PBPK model underpredicts the clinical DDI effect with digoxin. Now we performed a scaling of the in vitro KI value, and in order to be able to better predict the clinical DDI effect, we had to reduce the in vitro KI value by a very significant number, substantial number of 300-fold decrease of the in vitro KI to establish the in vitro in vivo extrapolation, which brings us to the graph on the middle. And here you see that the PBPK models predict much better the clinical DDI effect with digoxin for these 15 perpetrators that we used. And once this was established, so the scaling factor, then we applied that to the in vitro KI of our own drug, which, as you see on the graph here down, moved the prediction of the digoxin AUC ratio from 1.00 that we initially predicted with in vitro KI to about 1.37 at 40 milligram BID, which gave us weak effects at 40 milligram BID and 80 milligram QD, and then at weak effect and then moderate effects at 200 milligram BID. So the PBPK then predictions were submitted to the FDA as response to this information request, and our approach was accepted, and this also ended up in the label. On another query that we had during the regulatory review, so FDA asked us as a post-marketing commitment or post-marketing requirement to investigate what is the effect of a proton pump inhibitor in the exposure of asciminib at 80 milligram and 200 milligram dose level. And in order to do that, we also used a PBPK modeling approach, where we had in vitro dissolution data from two-stage experiments at 40 milligram and 200 milligram that you see here with blue and orange respectively in the fasted state, and then we had additional dissolution data from two-stage experiments with hypochlorhydric media, again at 40 and 200 milligram. And this information, along with particle size, solubility information, were used all together to develop another model within SimSip, where it was using 2 solid states to handle for the hydrochloride salt that the drug was formulated to, and also with modeling of the in vitro data within SIVA. And what we predicted was that the absorption of asciminib between 40 and 200 milligram in fasted state is predicted to be complete. And in addition to that, there was predicted no clinically relevant PPI effect at 200 milligram or 200 milligram or 80 milligram. And the PBPK modeling approach was accepted by the FDA, and the post-marketing requirement for additional PPI trial was also lifted. Now, some other interesting findings we have included here is about, on the left, for the fat-dependent negative food effect that we have significantly observed with this drug. So, as you see here from the publication by our colleagues, in the fasted state, we have the PK profile here, and then you can see the low-fat fat PK profile as well as the high-fat fat PK profile. So with, there's a reduction about 30% in AUC with the low-fat fat, and with the high-fat fat, so there's a typo here, about a 70% reduction in the presence of a high-fat fat meal. In addition to that, another observation we had, which was first to our knowledge observed by Genentech, was also a reduction in the exposure of asciminib in the presence of a cyclodextrin solution, which in our case was also, it was part of the itraconazole solution, which contained 8 grams of HB beta cyclodextrin. And there was a reduction of about 50% in the exposure. So if we move to the next slide, we'll see the challenge that we had and still have in predicting mechanistically the negative food effect for this drug. We have done significantly progress with the use of the ADA model. However, there are still some challenges in the difference in the presence of the fat content, as well as the digestion, and as well as the difference between low fat and high fat conditions. And then with regards to the cyclodextrin, in addition to the other post-marketing requirement that I mentioned for the PPI effect, which we addressed, there is an ongoing post-marketing requirement by the FDA where we were asked to address and define a threshold beyond which the cyclodextrins do not affect the exposure of asciminib in a clinically relevant manner, and this has to be done for different types of cyclodextrin. So, and to do so, we're going to use in vitro solubility and flux experiments, as well as in vitro permeability experiments, and this will be coupled with PBPK modeling to predict at which dose of the different cyclodextrins that the exposure of asciminib is not affected. So in the last application I have included as part of this presentation was the extrapolation of the hepatic impairment effect on the PK of asciminib. So as you can see from the clinical study at 40 milligram, there was an increase of about maximum of 56% in the severe hepatic impairment in the exposure of asciminib when compared to healthy individuals. And here there was also some challenges in accurately predicting the hepatic impairment effect because of the complexity of the elimination pathways involved in the metabolism of asciminib. One of these was the incorporation of changes in the UGT2B7 abundance from a publication from Prasad et al. That we had to implement into the model in order to check whether it improves the prediction of the hepatic impairment. And of course, after doing these changes and incorporating the UGT2B7 changes in the abundance, still the PBPK model, even though it predicted the exposure in hepatic impairment reasonably well, within 50% more or less of the observed values. Still, it was not entirely, it was not considered to be accepted by the FDA. On the other hand, there was some additional changes that were not really considered within the PBPK model. One of these being potential changes in the bile salts homeostasis, especially in the severe hepatic impairment, which might affect the exposure of this drug, as it's very sensitive on the bile salt concentration. And also there were no changes in the abundance of BCRP implemented for the hepatic impairment. Now, an additional work by our pharmacometrics colleagues was also performed using PBPK analysis to address the hepatic impairment effect on the PK of asciminib, and eventually, considering the totality of the evidence that was presented at the FDA, there was no additional study required at 80 milligram and 200 milligram, of course, considering the safety profile of the drug. On the right part of the slide, you see some top-down adjustments that we attempted in order to improve the fitting of the PBPK model in hepatic impairment. For example, the initial model was not able to predict well, particularly the Cmax, and then after some top-down adjustments, either on the KP scale or on the distribution part of the drug, Cmax was a bit better predicted, but the distribution phase was not that well captured. So eventually this top-down adjustment did not really improve the PBPK performance for the hepatic impairment. All right, and if we move to the next slide, so this slide summarizes the impact of the PBPK model into the USPI from the FDA, as well as from the drug product label in Europe from the SMPC. So particularly for section 7 and sections 12 in the USPI, we see that the PBPK predicts DDI results for CYP3A4, CYP2C8, and CYP2C9 substrates at 80 milligram QD and 200 milligram PID. You see here down the label language of closely monitor asciminib by 200 milligram with strong CYP3A4 inhibitors, and then closely monitor and avoid certain CYP3A4 substrates at 80 milligram and 200 milligram BID when asciminib with asciminib dose respectively, and other examples with CYP2C9 substrate as well as PGP substrates. There was a detailed section in the USPI with model-informed approaches, and interestingly, the PBPK results were also shown as actual clinical data without mentioning the use of PBPK modeling necessarily. And also, of course, the PBPK modeling supported the regulatory review in terms of information request and post-marketing requirements. With regards to the SMPC, since in Europe only the 40 milligram BID dose was approved, there was a much smaller contribution to the PBPK model, but there was an interesting mention of the prediction of the absorption, the bioavailability using the PBPK models in the SMPC. So if we go to the next slide, which is practically showing the take-home message, at least for the USPI, this is on the left what we submit to the FDA. So we submit prediction of perpetrator DDI at 80 milligram QD and 200 milligram BID, as well as victim DDI at 80 milligram and 200 milligram. And then we had untested DDI scenarios at 40 milligram, and then hepatic and renal impairment untested scenarios at 80 milligram and 200 milligram. In addition to that, during the regulatory review, we had the PGP substrates question as well as the PPI effect, and all this ended up to the label without the need of us to run any clinical pharmacology studies and for either the dose of 80 milligram or 200 milligram, with the exception of a post-marketing requirement to have to run a 200 milligram study with a strong CYP3A4 inducer. And eventually this led to us replacing more than 10 clinical pharmacology studies by PBPK simulations, which was a huge success. And in addition to that, of course, we have currently ongoing post-marketing requirements and post-marketing commitments, 2 additional post-marketing commitments with FDA to use PBPK analysis instead of a clinical trial to establish first an in vitro extrapolation for BCRP and OATP inhibition, and second to find out what is the effect of, what is the dose of cyclodextrins that does not affect the PK of asciminib. The last slide shows, if we move to the next, the last slide shows simply the impact on the EMA and the UK labeling language. This is what I mentioned before, that EMA states that PBPK models predict that asciminib absorption is approximately 100%, while bioavailability is about 73%. And for UK, that was approval of 40 milligram and 80 milligram. QD, there is some additional labeling languages for the DDI at 80 milligram QD for both victim and perpetrator. And with that, I would like to thank all these people from the asciminib team, the PK Sciences, Pharmacometrics, and TRD team, and management support within Novartis, and many, many others that have contributed for the success of Semblix. And I would like to thank you for your attention, and I'm happy to discuss any questions that you might have.
Thanks, Yannis. That was really good presentation. As you expected, got a lot of interest and a lot of questions came through. So I'll try and get through a couple of them as quickly as I can. So one of them relates to that over prediction of rifampicin, and it was asked if there was any impact or FU gut, or what FU gut was used for prediction in that.
Yeah, so that's a good question. So the FU gut that was used was actually adjusted. The adjustment was also justified. Initially, we over-predicted the Cmax ratio of midazolam, so this was kind of midazolam DDI that was kind of an initial indication for adjusting the FU gut, and eventually the value that was used with multiple sensitivity analysis on the Cmax and the use ratio of midazolam, also with the victim DDI for clarithromycin and itraconazole, was a compromise value between all these 3 different DDI studies to kind of match, and it was reduced from 1 to 0.25.
Okay, thanks. I'm going to do a few questions on kind of the PK side, and then I'll hopefully do something about the regulatory side as well. So did you monitor the PK of the CYP3A inhibitors in the clinic rather than adjusting the KI values?
So we had not the full PK of the CYP3A4 inhibitor, we had only the trough values.
Okay. Is there any reason why we don't see any DDI with itraconazole?
Well, yeah, I think, well, that's kind of hard to explain, because originally we had, the itraconazole DDI with the sporanox solution, which is supposedly to give much higher exposures of itraconazole, but then we had the cyclodextrin effect, but still we are not very sure why with the capsule we see only this 4% increase, even though the levels of itraconazole seem to have reached the steady state.
Okay. In terms of the population side, which part of the model was changed to predict patient pharmacokinetics rather than healthy volunteers?
You mean between cancer and healthy volunteers?
Yeah, so I guess in the population that was used, were you using healthy volunteer in the simulation, or were you using cancer patient?
No, so for the healthy volunteers, of course, I was using healthy volunteers, and then for the cancer patients, we used both North European Caucasian and cancer patients as a population. The reason we used North European Caucasian to compare with cancer was because of the larger age range that we were able to give. But eventually we saw that there was not really big difference in the PK prediction between cancer and the North European Caucasian population. So eventually we used the cancer population for the simulation of cancer patients.
Okay. I think with just a limited amount of time, I'll just ask something about the understanding that PBPK modeling for NDA submission is generally a prolonged process. Did you identify any shortcuts or pathways that you could be safely employed during future NDA submission development?
I think a good strategy, in my personal opinion, is to proactively approach health authorities with your PBPK modeling analysis plan and discuss, the implementation and what can be done. I think they are much more open to discuss in advance than when you come to the filing and you have a PBPK model. I think, eventually a circuit is also, as more validated or verified you can have your model, of course, the better it is, because, this is no brainer, but it really needs to, predict well the PK in a good dose of range and provide extensive verification of your model.
Okay. And just a little bit about safety, how FDA concerns about drug safety risk with the 80 milligrams QD addressed?
Yeah, so in this stage we had a relatively flat safety profile, and the drug was safe up to 200 milligram BID, so it was relatively easy for 80 milligram QD to justify, a little bit more difficult for 200 milligram BID, which was approaching the higher end of the safety profile. And of course, this was also supported by pharmacometric analysis with exposure safety and exposure efficacy.
Thanks. Okay, just one more, I think, just to get something on the KI and scaling it for. In slide 21, the scaling factor was 0.003. Is KI different to vitro KI, and how did you come to that value?
Yeah, so first of all, the in vitro KIs were redetermined in the specific assay, right? So this like for specific assay and for specific lab. And then to come to this value, we used different scaling factors, and then we're looking at which scaling factor best predicts the range of the clinical DDIs we had with different perpetrators and digoxin as a substrate. And once this was the value that was kind of best predicting across the DDI values. I mean, of course, you can scan the whole spectrum, but it was done in a reasonable kind of pace of different values that we tested.
Sure. Do you mind just do one more question? I think it's interesting because repacanide is obviously a substrate for 3A, 2CA, and 1B1, OTB 1B1. So what was the FDA reaction to accepting the modeling results to inform labeling?
Yeah, so we had different simulations, right? From the clinical DDI with the 40 milligram, which was like kind of the net effect of inhibition of those three, plus more simulations showing the individual effects simply by switching on and off the different KIs. So if you have like only 3A4, 2C8, only 2C8 and 3A4, and all the different combinations that were used to simulate the effects. And since the overall effect was weak, then there was not expected the individual effect will lead to higher.
Mm-hmm. Thanks, Yannis. And then also thank you to everyone who's asked questions. I know I haven't got through them. I tried to get through as many as I could in that time, but I'll leave it with Yannis to follow up with some of these questions as well. I just want to thank Yannis again. And just before you go, just to mention that the next webinar will be being able to register on the members area on SimSip, and this is looking at PBPK models for MABs in pediatrics and pregnancy, and this is presented by Dr. Kate Gill from Certara. So thanks again to Yannis for your time and great presentation, and thanks everyone for joining us today. Goodbye.
Thank you.